So now that we've covered a lot of treatments for bacterial infections, let's move on and look at some treatments for fungal infections. Fungal cells are eukaryotic, so that presents some special problems of its own. The majority of chemotherapeutic drugs or antibiotics are designed to act on bacteria and are thus ineffective for fungal infections. Because of the similarities between fungal and human cells, most things that are able to treat fungal cells also will have toxicity to human cells. There have been a few antifungals developed for both superficial and systemic fungal infections, and they fall into four main groups. There's the microlide polyene antibiotics, the azoles, the echinocandins, and the flucytosines. Let's begin by looking at the polyenes. These guys bind to the fungal membrane and cause loss of selective permeability. Fungal membranes contain a steroid that's called ergosterol, as you may recall, and the human membranes don't, which is what allows polyenes some specificity to the treatment of fungus. However, the sterol in humans is still pretty similar to the ergosterol, so in some cases we'll find that the polyenes bind very lightly to those, so there's still some toxic effect. The most effective and versatile polyene treatment is amphotericin B. It's used to treat candida infections of the skin and mucous membranes, but can also be injected for systemic infections. The azoles are broad-spectrum antifungal agents. They inhibit ergosterol synthesis also. There's three main ones that are used. Ketoconazole is taken orally or topically, and it's for cutaneous mycoses, vaginal and oral candidiasis, and some systemic mycoses. Fluconazole is used in selected patients for AIDS-related mycoses. Clotrimazole and miconazole are typically ointments for infections on the skin, mouth, and vagina. The echinocandins inhibit fungal wall synthesis. Those include capsofungin, micafungin, and they can be used against candida and aspergillus. Flucytosine is an analog of the nucleotide cytosine, hence its name. It can be used to treat certain cutaneous mycoses, and often it's combined with amphotericin B for systemic mycoses, because many fungi are resistant to flucytosine alone. Now there's one more class that's only used topically. These are the griseofluvins. They're used for dermatophyte infections, so infections of the skin like athlete's foot. They're deposited into the epidermis, the nails, or the hair, and they inhibit fungal growth by binding to the microtubules and preventing further penetration into the tissues. So protozoans are also eukaryotic cell types. Again, they present a little bit of an issue with treatment. We'll look at Plasmodium separately. It's the malaria parasite, and there are several different species of this parasite, and there are many different stages in the life cycle involving several hosts, as you've probably seen on the wiki post. So no single drug is really effective for all species and stages of the life cycle. Malaria has been treated with quinine for decades, hence the development of tonic, which was originally brought around in the time that Panama Canal was being built, so that the builders could better manage some of the symptoms of malaria that they were getting from the mosquito bites. Interestingly enough, the gin part of a gin and tonic came along because it helped the tonic taste better. Anyway, that's the story that I understand. All the drugs today that are used to treat malaria are synthesized quinolones. And there are a number of different types used for a variety of different stages of the life cycle. Now, other protozoan infections have a few different drugs that we can choose to treat them. You may have heard of Flagyl or metronidazole. It's effective at treating intestinal infections like Entomoeba histolytica or Giardia lamblia. It's also effective for Trichomonas vaginalis, a sexually transmitted disease. Other protozoan infections can be treated by some of the sulfonamide antibiotics, as well as the tetracyclines. So this is an area where we see some crossover between treatment of prokaryotic cells and eukaryotic cells. You'll recall that the sulfonamides interfere with folic acid synthesis. and the tetracyclines are about protein synthesis, but somehow they act well in some protozoans also. Now, how about the helminths, the worms? These are the flukes, tapeworms, and roundworms. They also have great similarities to human physiology. So using drugs to block their reproduction is not usually successful in eradicating the adult worms because they're so similar to us. The most effective drugs in the treatments of helminths immobilize, disintegrate, or inhibit the metabolism of all stages of the life cycle. If we don't get them at all stages of the life cycle, they can still persist. Mebendazole and thiabendazole are broad-spectrum antihelminthic drugs that are used in several roundworm intestinal infestations. They work by inhibiting the function of the microtubules in the worms, as well as the eggs and in the larvae. Without microtubules, we really disrupt cell structure as well as the mechanics that control cell division. Finally, we'll consider some antiviral chemotherapeutic agents. Selective toxicity is almost impossible to achieve because single metabolic system is responsible for the well-being of both the host and the virus. After all, the virus is taking over the machinery of the host to make proteins using its machinery, but the host still needs that machinery in order to make their own proteins. And nucleic acids are at the core of the whole thing. But there are several antiviral drugs that have been developed. They target specific points in the infectious cycle of the viruses in order to allow a little more specificity. There are 3 major modes of action. We can bar the virus from entering the host cells by taking away or hiding some of the access points that it uses. We can block transcription and translation of the viral molecules so no more viruses can be made. And we can prevent the maturation and release of those viral particles. Now, none of these methods are 100% effective. Otherwise, we would be able to get rid of viruses. But as you know, viruses are very tough to remove, especially when you consider HIV and AIDS. Here are a few major antivirals, just to give you an idea of how some of them work. The table on the right will help you understand some of these different mechanisms. However, we'll be exploring some of them in more detail later. Amantadine, for example, inhibits the replication of influenza A virus, so they can't replicate themselves. Acyclovir is used in herpes viruses. It disrupts their replication. Ribavirin blocks RNA synthesis, again, interfering with virus production. There are a few more details over here in the table, but you don't need to understand them just yet. Finally, there's an alternative to artificial drugs. Interferon is a human-derived product, and it's showing up to be quite effective in a number of different means. It has both antiviral and anti-cancer properties, and it's produced by recombinant DNA technologies. That is, we've taken the gene from the human genome and we've placed it into bacteria or some other production facility in order for more and more interferon protein to be made. It's a glycoprotein. It's naturally produced by the fibroblasts and leukocytes in response to various immune stimuli. It reduces the time of healing of some of the complications of certain infections. It can be used in preventing or reducing some symptoms of cold, as well as the papillomavirus. It also has been shown to slow the progress of certain cancers. However, at this point, we're still finding that it results in some pretty serious side effects. Let's take a quick look at a video to see how it functions. We'll be exploring these mechanisms further when we get into a later chapter this semester.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
So this is a pretty cool technology that we have stolen from the immune system and are now using biotechnology to make this interferon protein so that we can better fight some viral and cancerous invasions. Before we move on, let's have a quick concept check. Which of these antimicrobial compounds would best treat a severe genital herpes outbreak? Well, again, just checking to see that you're awake. Acyclovir we just covered. The other three of these are involved in AIDS treatments. An unfortunate outcome of our increasing use of antimicrobials is the development of resistance. So let's spend a little time thinking about what drug resistance is and how it happens. Drug resistance is an adaptive response in which microorganisms begin to tolerate an amount of the drug that would ordinarily be inhibitory. This resistance can either be intrinsic or acquired. Intrinsic resistance comes about because bacteria that produce antibiotics need to have resistance to the antibiotics that they produce. So that makes sense. Acquired is the kind we're really worried about, where we had previously sensitive bacteria that are now resistant. So that's going to be the focus of what we're covering when we consider drug resistance. Microbes can acquire resistance through either spontaneous mutations. They can acquire an entire new gene or set of genes via transfer from another species. These are called resistance factors or R plasmids. New evidence suggests that some bacteria can even go to sleep when exposed to antibiotics, shut down all of their metabolic processes so that they can later wake up when there's less concentration of the antibiotic. So let's look a little closer at R factors, because this is what really concerns us with the overuse of antimicrobials. You'll recall from a couple of chapters back, we talked about the horizontal transfer of genes from one bacteria to another. And this can happen between species, it can happen between viruses. The terms conjugation, transformation, and transduction should ring a bell. This is how we get different pieces of DNA, whether it's plasmids or pieces of the bacterial chromosome, between different bacteria in order to effect genetic variation. Because simple cell division doesn't really acquire any genetic variation. Sometimes we'll see that there are factors lying in wait for an opportunity to be expressed or to confer adaptability. So they're silent, and they may become active when the appropriate environmental cue arises, like the presence of an antibiotic. We can also have transposable drug resistance sequences or transposons that have resistance factors. They can be replicated and held within the bacterial genome, or they can be inserted into their own plasmids. We call these plasmids R plasmids. These pieces of information can be inherited by the subsequent progeny, and we'll see rapid proliferation of drug-resistant species, simply from just passing these resistance factors from the parent to the offspring. Now, R plasmids are where the resistance factor gets wrapped up in a plasmid, and it can be transferred through the process of conjugation. This can happen even in our own biota. So let's say the flora and fauna that are desirable in our intestines have resistance plasmids, then they can pass them into pathogenic bacteria. That is, those resistance factors can literally jump from our flora and fauna into the pathogenic bacteria. Now, this is a problem. How might you get a load of resistance plasmids in your intestines? Well, with overuse of antibiotics, not only are we conferring resistance to the pathogenic bacteria, but also to our own each time we use those antibiotics. Eventually, we rebuild our own biota in our intestines, and now those bacteria have the R plasmids. So it's super important that we only use antibiotics when we really need them. Let your immune system fight, because it'll make your immune system stronger. And then when the infection is just too much for your immune system to manage, then it's a good time to use the antibiotics. Here's another scary thought. Recently, scientists have learned that a wide variety of soil bacteria can survive the presence of antibiotics, even to the extent that they'll use the antibiotics as fuel. Now, those bacteria in the soil can just as easily pass resistance factors to disease-causing bacteria. So just think about our widespread use of antibiotics and how fast we're passing resistance factors around. Here's a few of the mechanisms of action for resistance. Consider that the R factor might code for some of the proteins involved in either of these mechanisms. So first of all, we've already explored the idea of penicillinase. Here we have active penicillin, but if the bacteria can produce penicillinase, which is a protein coded for by nucleic acids, which could exist on the resistance factor, then we can inactivate the penicillin so it can no longer have the same effect on the cells. We might also see decreased permeability. You have normal receptors shown over here. Now, if there becomes a mutation in the DNA or an R factor that now codes for a different receptor protein, the antibiotic cannot even enter the cell. Another strategy would to be activate drug pumps. These are specialized membrane proteins that are activated and continually pump the drug out of the cell. Again, a resistance factor could code for this protein. Another strategy might be to change the drug's binding site. If the binding site on the target, for example the ribosome, is altered, the drug can no longer affix to it and thus there'll be no effect. Perhaps the antibiotic knocks out enzymes along this pathway, but the resistance factors can actually code for different proteins that allow conversion of the same products through a different metabolic pathway to end up with the end product. So what's the big deal if some of our normal biota get antibiotic resistance? Well, let's look at it this way. Any large population is likely to have a few cells that are already resistant, just as we're showing here on this petri dish. If there's no drug present, this population will stay in low numbers because they have no particular advantage. However, if the population is exposed to the drug, all of the sensitive individuals are wiped out. All that's left to reproduce are the resistant strains. Now they have a selective advantage. They're going to survive preferentially and proliferate. In subsequent generations, we have more and more and more of the resistant form of these bacteria. All that's left in our petri dish are resistant forms. So in this fashion, a whole population can become resistant. How do we prevent this rapid spread of resistance? Again, limiting drug use. That means there's less selective pressure and thus less resistant bacteria are formed, even in your own intestines. Proper drug use. Anytime you have some kind of sickness, keep in mind that viruses are not going to be affected by antibiotic use, only bacteria. So there's no sense in just cranking on the antibiotics, because if it's a viral infection, we're not going to feel better. All we're going to do is promote resistance in our internal environment, in our good bacteria. If you do choose to use antibiotics, make sure that it's a narrow-range antibiotic. That way, we only kill the targeted microbes and only select in a smaller population. So only that specific type of bacteria are likely to become resistant. There are also less likely complications when we target a narrow range of pathogens. Also, using synergistic drugs, so multiple drug treatments, can mean a much lower dose and thus much less likely that resistance is encouraged. These are key things to keep in mind if you get sick. Also, you might consider the amount of antibacterial product you use. It does the same thing. If you're spreading antibacterial product all over everything and cleaning all surfaces with antibacterial product, then you're selecting for resistance forms. So the more antibacterial product you use, truly, the more dangerous your kitchen could become. Now, in recent years, people have been thinking about controlling infections before they happen through the use of probiotics and prebiotics. Probiotics are preparations of live microorganisms, for example, yogurt, that will improve our intestinal biota. Perhaps you want to take a different approach than taking antibiotics. Maybe we can help nature along by using probiotics and prebiotics to control infections before they arise, also allowing our immune system to become as strong as it possibly can. Probiotics are preparations of live microorganisms that improve our own intestinal biota. Yogurt is a great product. They can either be used to replace microbes that are used in an antimicrobial treatment, so following consumption of antibiotics, but we can still use them every day to augment the biota that are already there. It's now been suggested that many of these probiotics are great treatment for food allergies, and that perhaps they stimulate our mucosal immunity, which means we're boosting our own intestinal immunity so that we can take care of any invading pathogens. You can buy many of these products as supplements. However, they really should be refrigerated. If you think about it, they're live microorganisms, so the age of the product really matters when you're looking at probiotics. Prebiotics are substances that encourage the growth of beneficial microorganisms in the intestine, and they also may discourage the growth of potential pathogens. So knowing this information and how detrimental in the long run that the use of all these antimicrobial products as well as antibiotics could be to the health of not only your family, but the whole population in general, perhaps you'll think about how necessary it is to spread antimicrobial products throughout your environment, because all that does is allow for more rapid selection of resistant microbes, and we don't want that.